Okay, thank you, Javier, for the introduction. So again, I am Katia, and today I will be describing some of the highlights of the results obtained by using multi-omics approaches to deep insight into the complex heterogeneity of cancer diseases in the search of driver genes with clinical relevance. So as a bit of context, cancer is a major health issue. So I will present some fast facts. Millions of people are diagnosed with cancer each year. Cancer is among the leading causes of death, is one of the major killers worldwide. It is remarkable that 1 in 3 people will have cancer in their lifetime, which of course represents a very economic important burden invested in cancer care. So as a consequence of the importance of cancer in public health, multiple efforts have been developed to identify new therapeutic molecules to ameliorate medical care. So in this context, the oncology practice has incorporated key genes that are driving tumorigenesis as biomarkers to improve patient diagnosis and to design better therapies. So, however, to date, tumor heterogeneity remains as the most challenging feature in the diagnosis and treatment of these diseases. So, in this context, we focus on studying the significant heterogeneity of tumors at systems biology level, emphasizing the potential clinical of driver genes towards personalized medicine, where in the future a patient could receive the right therapy. So for this purpose, we have integrated data from genomics, transcriptomics, and proteomics datasets that are publicly available and, developed and implemented bioinformatics pipelines to differentiate between healthy and diseased cell samples. So cancer research can be summed up in a single sentence. Cancer is, in a sense, a genetic disease. So progressively, there has been accumulated knowledge on those driver genes that are having potential relevance in clinics. So we took advantage of this, and using data mining of literature, we identified 18 genes that are driving tumorigenesis in a very heterogenic cancer that is glioblastoma. And we clusterized them according their functional in cytoskeletal and proliferative genes, tumor suppressors, and also DNA repair and chromatin remodeling. So using transcriptomics analysis, we identified a transcriptional profile pattern in glioblastoma that fits with the stem cell model of ontogenesis. And moreover, we also identified 4 potential biomarkers with the clinical relevance to differentiate among the glioblastoma molecular subtypes, which of course could be helpful to delineate in a future and determine which treatment a patient should follow. We also study the somatic mutations and focus on analyzing the biological relevance of mutations using the polyphen impact. The polyphen impact is a measure of a mutation consequence that could be probably damaging, possible damaging, or benign. So we cluster driver genes according patient age and identify that in the young subgroup, when compared to the other ones, the DNA repair and chromatin remodeling genes are undergoing different somatic affectations. So moreover, we identified that OLIC2, that is VHLH transcription factor, is only affected in the early group. So moreover, we deep insight to the better understanding of the behavior of these mutations in patients, analyzing and focusing on the most lesion genes grouping by patient age. So we identified for the young patients a different regulation of undergoing for transcription factors, and that the enrichment biological and molecular processes are different from those that corresponds to the other patients. So moreover, we identified that the affected genes are fewer conserved in this group. So since cancer is a genetic disease where genes are undergoing disruptions, we later focused on a study where we were interested in in the clinical potential of a very particular subset of genes that are intronless genes. Intronless genes are single exon genes that are nuclear and that have coding capacity and codify for functional proteins and that lack introns anywhere in their genetic structure. So intronless genes could be potential biomarkers since we previously identified that some of them are highly expressed, and of course that they exempt alternative splicing. And for that reason, they could be undergoing faster genetic modulation and also have a very predictability as biomarkers and easy regulation as targets of therapies. So for those reasons, we believe that X could have a potential as clinical biomarkers and drug targets that deserve careful consideration. So in this slide, I presented, I want to highlight the role of one of X that we use in a clinical report, which is SOX11, that is a biomarker to differentiate among non-Hodgkin lymphomas. So in this clinical report, we identified that the disease diagnosis of this patient that presents erythroderma is a paraneoplastic manifestation of mantle cell lymphoma instead of T-cell Sézary syndrome. So we can note in this context the relevance of translational research, of course, of biomarkers. So as an objective, we want to identify their unique expression profiles and interactome that may act as functional signatures across different cancers. So first of all, we identified 940 human intronless genes that are enriched mostly in DNA repair and chromatin remodeling processes. And moreover, intronless genes have tissue specificity when we analyze in healthy samples, tissue healthy samples. So we use TCGA data. Basically, we analyzed the 4 most prevalent cancers, which are breast, lung, prostate, and colon. And we also selected 4 of the most highly with severity heterogeneity, aggressive and invasive cancers, which are namely bladder, esophageal, glioblastoma, and kidney. As an overview of the comparison of this subset of genes against the multi-exonic ones, we identified that X tend to have higher expression range compared to those of the multi-exonics. So moreover, in this pan-cancer study, we identified that 35% of them were differentially expressed across the analyzed cancer datasets. So glioblastoma, followed by kidney and prostate, displayed the highest specificity for this subset of genes in their the regulation. And interestingly, almost 80% of them are undergoing transcriptional reprogramming with elevated expression in tumor cells. And they are encoding histones involved in negative gene regulation. So to deep insight into this, we also analyzed that a highly conserved induction of a particular group of the acetylasa histone located in the region 6 of the human chromosome is conserved among the cancers and is enriched in nucleosome and chromatin condensation processes. So also we use network approaches to study cancer, and we identified that the differential expressed X across cancer types are prevalent in epigenetic regulatory roles, participating in specific protein-protein interaction networks for esophageal, glioblastoma, and lung tumors. And also we identified that our cancer-specific differentially expressed X that are encoded proteins interact with specific groups of proteins in distinct cancers. So what I'm showing here is the 8 analyzed different tumors, and what you are seeing in highlighted in yellow is a differential expressed intronless genes that is interacting with particular specific proteins in the different contexts of the tumors. So finally, we also analyze that X play a key role in the tumor phenotype, not only at the transcriptional, but also at the post-transcriptional level, and participated in important mechanisms such as rewiring. So basically, histones play a key role in this mechanism from the healthy to cancer tissue. So summarizing, we study a very high intrinsic heterogeneity tumor, which is glioblastoma, and identified transcriptional profile patterns fitting with the stem cell model of ontogenesis, and detected 4 biomarkers that have high potential relevance to determine a particular molecular subtype of this tumor. And we highlighted the potential regulatory role of the differentially expressed human intronless genes across this pan-cancer study, and identified that that they're participating in specific protein-protein interaction networks for glioblastoma, esophageal, and lung tumors, which are also a highly intrinsic heterogenic environments. And finally, we identified that they are particularly important in mechanisms such as interactomic rewiring using breast cancer data. So if you are interested in more information or more detailed information of these studies, you can scan the QR codes and go directly to links of the publications, and also to the lab, to the link of the lab that has been mainly working on this project, which is Maribel Hernandez lab. And I want to appreciate the work of my co-authors and people that has been enrolled in the development of this project. Also, CONACYT and Fulbright for providing the funding for my fellowships, and Reavio for providing the travel grant to attend this conference, for also Simvestab and the University of Illinois, where I am basically working. And I invite you to go to the poster hall to where there is a video that Tonio left, where you can notice and more information about the rewiring analysis in breast cancer. And of course, thank you to all of the team, and thank you to the audience and to the organizers. And that will be all for today. And I will be happy to take any of your questions.